Yes.
Okay, sweet. So we're just going to go over a few things with the immune system and the lymphatics. So we'll start off talking about primary versus secondary lymphoid organs. So primary lymphoid organs being where lymphocytes mature, and secondary organs being where they actually go and attack any different antigens and basically cellular debris and stuff. So primary lymphoid organs, we have the bone marrow and the thymus. So although B cells and T cells, which are two different types of lymphocytes, they both develop in the bone marrow. The B cells mature. Oh, that doesn't work. The B cells mature in the bone marrow, whereas the T cells mature in the thymus. So I'll write that down. So B cells, they mature in the bone marrow, and then the T cells mature in the thymus. So the thymus is highly active in the first year of life and up until about puberty, and then after puberty it slowly starts to degenerate. And it becomes almost indistinguishable between the thymus and the connective tissue surrounding it as we move later into life. Because we don't need to develop more T cells, we have them all set. Secondary organs, that's where our B cells and T cells actually encounter antigens. So secondary organs are the
spleen.
Yeah, is this the spleen? Yeah, the spleen, the lymph nodes, and the mucosa associated lymphoid tissue?
Yeah.
So spleen, lymph nodes, and mucosa associated lymphoid tissues. So the spleen basically filters the blood, and then it stores platelets and monocytes. The lymph nodes, they process debris found in our lymphatics, and then mucosa associated lymphoid tissue serve basically immune responses in our mucous membranes. So we'll move on from that, and if you guys have any questions, you can leave them for us, or just read Marie, because that's where it all came from. So, different lymphoid cells. So we have macrophages, lymphocytes, and reticular cells. So within the lymphatics, these are the kinds of cells that are going to be found. So macrophages being our phagocytes, lymphocytes being our adaptive immune cells, and then reticular cells being the cells that actually provide connective tissue support within all of our lymphoid organs. So let's move on to innate and adaptive immune system. So the innate immune system, this essentially is just all of our surface barriers and then our internal defenses. So surface barriers being the skin and the mucous membranes, they secrete acids, different enzymes, things of that sort to break down any pathogens that are on our skin or get inside into our mucous membranes. Mucous membranes also secrete something called mucin, or mucin, I don't know how it's pronounced, but when it's mixed with water, it actually forms mucus, and that is so that we can trap different pathogens. We can either export them through the body, so get rid of them with sneezing, coughing, stuff like that, or we can swallow them and digest them. Then as we move to our internal defenses, so we have phagocytes. This is found in both the lymph and the blood. Then we have natural killer cells. These cells are far less picky than lymphocytes, so they typically find cells that are infected by viruses or cancer cells before our adaptive immune system is alerted, and they'll signal them to undergo apoptosis, which is programmed cell death. Then we have antimicrobial proteins, and those are, we have interferons and complements. So I'll explain a little bit about them, but not too in depth. Again, there's more in the book, and if you have questions, you can ask us. But interferons, essentially, if a cell is infected by a virus, so the virus somehow gets in there and infects the cell, and the cell is now creating nasty proteins within it, interferons will come about in the cell, and they will actually see that this cell is infected, leave this cell, and go to the cells around it, basically telling other cells in the vicinity there is a virus. So be prepared. So then this cell can do a bunch of stuff to prepare for any kind of virus infection. Complements, on the other hand, they're found primarily in the blood, and they're proteins that can actually lyse microorganisms. So any microorganisms in the blood, complements can either take care of them, or they mark them for destruction by lymphocytes or macrophages. So they'll either kill things, or if they can't, macrophages and lymphocytes will take care of them, and complements will essentially mark them for their destruction. So let's move on to adaptive immune system. This one's a little bit more in depth, and I'll try to cover everything, or at least most of it. But we have cellular immunity and humoral immunity. So cellular immunity is the you can erase that. Cellular immunity is the adaptive immune system response within our cells. So intracellular. And then humoral is basically referring to the humors of our body, humors being fluids. So this is within blood and lymph. And then interstitial fluid, so outside the cells though. So extracellular. Okay, so we'll start off with APCs. We'll talk about T cells, B cells, and then we'll talk a little bit about CD4, CD8, MHC1, MHC2. So APCs, these are antigen presenting cells. I'll write it down. And antigen presenting cells, we have three primary antigen presenting cells. So we have macrophages, we have B cells, and we have dendritic cells. Dendritic cells, we likely have less dendritic cells than B cells and macrophages, but their only job is to be an antigen presenting cell. So antigens being substances in our body that we recognize as foreign. So we recognize them as a non-self. So these cells will collect those. So let's say this is a B cell, for example, and there's an antigen outside the cell. Okay, that's the antigen. We will have basically these little receptors on the membrane. They will bind to that antigen, and they will signal to T cells that we have an antigen. Now, there's a couple different ways that we can do this, and this has to do with MHC1 and MHC2. So, MHC2 recognizes extracellular antigens. So these kind of antigens. Basically, MHC2 brings these antigens inside the cell. So now the antigen is inside the cell. Let's say it's bound to that MHC2. And it will digest it a bit. It will bring it back to the surface. And it leaves it there. And it's a signal for T cells to come deal with it. So in this case, this being our B cell, which was our antigen presenting cell, MHC2 bound to that APC, brings it inside the cell, digests it a little bit, brings it back to the surface, signaling for a T cell to come help. In this case, being that it's MHC2, we get a CD4 T cell, which comes to its aid. So CD4, this is our CD4 T cell. Okay, so CD4 comes and it recognizes that this is the bad guy. CD4 cells, we see them in the form of helper T cells. So helper T cells meaning that they don't actually deal with these antigens, they signal for help to B cells. So in this case, this T cell, this CD4 T cell, the helper T cell, finds this antigen that was presented by the B cell, and now it will actually, now that it sees this antigen, it will divide into effector cells and memory cells. The memory cells are for our long-term immunity. So these ones aren't actually active in fighting these antigens. They just get stored essentially in our lymph and blood, and yeah, they are used as memory cells. So that way, if we re-encounter this antigen, this virus, whatever it is, if we re-encounter this again, our body recognizes it sooner and can deal with it quicker. In that way, in that sense, if we get sick, let's say with a cold, we'll never get that same cold again. So every cold that you've ever had, it's never been the one that you had before. It's a different virus. Now the effector cells that we have are helper T cells. So same guy here, but this is now an effector helper T cell. It will secrete these little things which actually activate B cells. So these things go over here and they activate this B cell, so this B cell can now secrete antibodies. So now this guy secretes, let's just say, these, I don't know, rounded shapes here. That's a square with a triangle. These are our antibodies, and they will go and deal with not only the antigens that our B cells have, but anything within that extracellular space. So whether that be in the blood, lymph, or like interstitial space. Okay, so that's kind of MHC2 CD4, and that's kind of how it works. If you have more questions, just let us know, and I can try to do a better example of that. Now we'll talk about MHC1, because this has more to do with intracellular immunity. If you want, you can erase that. So, MHC1 being intracellular immunity. So just like we had that antigen which was outside the cell, and it was recognized by a B cell or antigen presenting cell, so it could have been a dendritic cell, it could have been a macrophage, doesn't matter. It was recognized, and MHC2 brought it inside the cell, digested it, brought it to the surface. We can have viruses or other things within our cell that are antigens that we recognize as non-self. So for some reason, let's say when we're creating specific proteins within the cell, something happens and we make something that's messed up, and it's, let's say now this is a cancer cell and it is infected. So the cell is infected with a bunch of these shitty proteins, and now this thing is going to divide its cancer cell, and so we're going to have a ton of these guys. What happens is we have MHC1 in the body, so let's say this is MHC1. It will go and it will bind to these antigens, present them to the surface, just like in the first case, except now these antigens are starting in the cell, and we will have a CD8 cell bind to it, and we'll say in this case, this is our CD8, it will bind to it, and it will do the same thing, it will divide. So we'll divide into effector cells and memory cells. Effector and memory. Same thing applies as before. Effectors are the ones that actually do the work. Memories are the one that provide us with that long-term immunity.
Is CD8 a B or a T cell?
CD8 is a T cell, but in this case it is a cytotoxic T cell. Okay? So it divides into cytotoxic T memory cells and cytotoxic T effector cells. So now we have these effector cells. What they will do is they will go and they will bind to the cells that are infected with these antigens. Again, MHC1 is presenting these antigens to the surface, and they will actually send chemicals into the cell to destroy these antigens. And so that's how we deal with that. If you get confused about remembering MHC1, MHC2, CD4, CD8, yes, we talked about this in class, but MHC1, okay, that one is coupled with CD8, in this case, cytotoxic T cell, MHC2, CD4, helper T cell. And to remember that, you can just remember that the answer is always 8. 1 times 8 equals 8, 2 times 4 equals 8. But that is there. So again, MHC2 deals with extracellular antigens, MHC1 deals with intracellular antigens. That's pretty much it. I think that's all we'll go over for now. And if you guys have questions, just let us know or read the book because it's all from the book. Sweet.
Say bye.
Bye.